All right, I think we'll get this started, and people can continue to trickle in and join as they're able, but I want to start by saying hello and welcome to everyone. This is the Huntington's Disease Society of America Research Webinar Series, and we are excited to have Dr. Jane Paulson with us today, talking about the PREVENTHD study. Kelly, if you can flip to the next slide. Just a little bit of logistical information as we get started. We are going to offer an opportunity for people to ask questions and have them answered. That will happen at the end of the presentation, but we want you to know that you can submit a question anytime over the course of the presentation through the chat function that's located on the toolbar at the bottom of the Zoom screen. So if you click on the chat function, it'll give you an opportunity to type in your question. Just type it in and hit send. The only people who are going to see the question that you submit are those of us who are monitoring the tech part of it. So it'll go to us at HDSA, and then we'll present those questions to Dr. Paulson at the end so that she can answer them. So feel free to enter those at any time, and we'll be looking for them as they come in. We also want to let you know, as you may have heard at the start of the session, that the webinar is being recorded. So if you don't catch it all, if you come in later or you weren't able to make it at the time that we're hosting it live, you can absolutely find this webinar on the HDSA Research Webinar site. That is www.hdsa.org/researchwebinar. Or you can find it on our YouTube channel, which is a link on the hdsa.org homepage. And something that we want to add, this is a very timely matter for those of you watching live or hopefully in the next couple of days. The HD Parity Act is something that we have talked about for a number of years now, but is something that is sitting at a kind of important time point. We've had a very successful advocacy week about the end of October. We need some targeted legislative push here from people in the community. And you may be tired of hearing this because it's been going on for it's going for a lot of years, excuse me. But right now we're at a place where there is more action happening. So if you are a resident in the state of California, Massachusetts, New Jersey, New York, or Oregon, we're really looking at you to put some pressure on your representatives. You can find this by going to hdsa.org/takeaction. It will provide you all the resources you need to get in touch with your offices. If you don't live in those 5 states, of course, your voice is important too. The more noise that we can make right now, the more people we can get sharing their stories, the more we think this can actually push the HD Parity Act over the finish line. Again, you might say, we've been hearing this for years, but right now we're just asking you to give this extra push, especially if you're a member of those 5 states. So just wanted to put that out there. Take a look at it if you can.
All right, just a reminder, if you are interested in participating in research studies like the one you will hear about today or others, you can learn about them on hdtrialfinder.org. It's a website for you to match to clinical studies in your area and nationwide, and to stay up to date on the research in the pipeline, both in the United States and across North America. For other resources to learn about HD research and to participate, you can check out hdbuzz.com. It's a group of HD scientists who write articles about Huntington's disease research, as well as HDSA's research blog, which you can access through our website at hdsa.org. Also, if you'd like to participate in research, Enroll HD is a great ongoing study. You can learn about Enroll and others on HD Trial Finder, as I mentioned before, or through a center of excellence near you. And all of this information is available again on HDSA's website. And with that, I'll hand it back over to Eric to introduce our speaker. Oh, just kidding, sorry. Upcoming HDSA webinars next week, we have 2, a disability webinar with our resident disability specialist, Allison Bartlett, as well as an HDSA webinar next week on Thursday, focus on HD advocacy with Jennifer Simpson. And our next HD research webinar will be next month, December 7th, with the editors of HD Buzz. It'll be an open Q&A with the community.
Awesome, Kelly, thank you so much. I am very happy to have the privilege to introduce our speaker today. Today we'll be hearing from Dr. Jane Paulson, professor of neurology at the University of Wisconsin, and the lead on the PREVENTHD study, focusing on preparing for prevention of Huntington's disease. Dr. Paulson's research interests in neurodegenerative diseases began in graduate school, and she initially published in the disease areas of Alzheimer's and Parkinson's. Her work in Huntington's disease was led to by a curiosity of the region of the brain associated with motor control, which is the basal ganglia. And since that time, she has worked with all movement disorders as well as other neuropsychiatric diseases of the basal ganglia. Dr. Paulson's passion for early detection of disease increased with the sequencing of the human genome and the detection of the gene that causes HD. Dr. Paulson created the first natural history study of gene expansion carriers for HD, when there were nearly 500 investigators collaborating to detect the biological, imaging, and phenotypic measures of the earliest signs and symptoms of the disease in 1,500 people from 32 sites and around the world. This was the PredictHD study. Dr. Paulson continues her work to detect and track early measures of HD in HD gene carriers in the PREVENTHD study, which continues to refine and develop methods to prepare for preventative trials in HD. Dr. Paulson's collaborative team science encompasses a broad range of neuroscientific arenas, including imaging, phenotyping, biomarker discovery and development, as well as empowering participant efforts to address discrimination and stigma in genetic brain disorders. Finally, Dr. Paulson is passionate about teaching, training, and mentorship to expand the field of scientist practitioners who can more readily move discoveries for our patients and families through transitional neuroscience to keep the research initiative going for HD to get to a place where we have better treatments and interventions available for our patients and families. So Dr. Paulson, thank you so much for being here and for talking to us today.
It's certainly my pleasure. And now do I take the slides sharing over? Is that what I do?
Absolutely. Go ahead and share your screen.
Okay. Okay, so let me know if something doesn't go well, because I'm not sure I always know. So feel free to poke in and let me know. First, I just want to thank HDSA again for just being present throughout my whole time I've been working with Huntington's disease, and really trying to bring us all together and being a shared resource where we can all go to try to make sure that we're doing the best we can for those impacted by Huntington's disease and ourselves, and that we continue to move the science forward while we take care of each other. And I just want to thank Drs. Fox and our newest Drs. Johnson today for hosting these webinars and keeping all of us together on what's going on. And I think we just need to keep doing this as much as we can and encourage one another to work together, because I think it's this teamwork that's really going to help us move the progress forward. So I first developed this slide, or borrowed it from someone, literally 21 years ago, when we had the PredictHD study funded, because it was the first study to really look at people with Huntington's disease genetics who did not manifest symptoms. And at that time, there was a lot of pessimism about whether this study could even be done. And I think because of the partnership of the families and the scientists, and really the cohesion that has worked among all of the organizations through other organizations like HDSA, but just across the world, we've really learned a lot in the last 20 years. And I think it's a good time for us to remind ourselves how much we've learned and how much we've grown in these past few decades. It was certainly disappointing last year when the 2 genetic therapies for Huntington's disease, slowing progression or disease modification, had to halt. But I think it's even more important for us to get together on the same page again and get a renewed interest. There's so much to do. There's so much that's been done, and there's so much left to do that we can't allow a slow up. We can't allow COVID to get in our way. We can't allow our disappointment to get in our way. We really need to be there for one another to keep things moving forward. Gotta make sure I can advance that. Okay, so today I just want to focus again on what do we need to do next? Because for Pete's sakes, this community has been amazing at advancing our knowledge of pre-diagnosed Huntington's disease. We had, as you know, 1,500 people in PREDICT. We had 300 or 400 more in TRACK and TRACK On. Now we have thousands and tens of thousands in Enroll, and we're really all trying to put our best foot forward to make a difference. But I think what we need to do is figure out what do we need to do next so that we don't allow any challenge that happens in life constantly to slow us down. So I'm just going to start with the requirements from the Food and Drug Administration. This is the overall governing body for the United States that determines what treatments we can give for what purpose to what patients. And they not only approve the treatments, but they also give us guidelines for how to keep progressing our medical care for all diseases, including rare diseases like Huntington's disease. So these are the first 3 requirements they have if we want to move into a prevention framework. That means looking at disease treatments, not only once you have a motor diagnosis of Huntington's disease, but earlier than that, when you may have more subtle manifestations of the disease, or you may still be able to keep your work, raise your kids, enjoy your life, but there's some things going on. What can we do to work on that a couple of decades before diagnosis? As we all know at this point, HD is a slowly progressive disease over what we now believe is probably more than 40 years, because we'd already known from the great scientists before us that the average age from motor diagnosis to death ranges from 17 to 20 years. And now we know from the PREDICT study that we can see changes in a person 20 years prior to motor diagnosis. So we really need to keep working together to advance what we do with this knowledge. So these 3 questions is, first of all, the FDA is going to say, do you even know about this disease? Give me the natural history. Do you know what happens without a treatment? Because they don't want to have us start too quickly in treating patients if we really don't understand what's going on. The second question is, do we really know how to diagnose and identify this disease so that we're testing the right people and we're giving the treatment to the people that can most benefit? And the third question the FDA is always going to ask over and over is, can we really get the volunteers in for these clinical trials? And the reason you hear these same questions over and over is because even though we answer them, they're still important. So that is why it's all more important for me to just get the message out today that we can't give up, we can't slow down, we need to pull back together and get everyone working towards, in this new era of post-COVID, post-Roshan wave, first generation ASOs, what are we going to do? Well, we're not going to slow down. And that's why I need your help to figure out what can we do to keep things moving forward. So to me, the first question, these are 3 slam dunks for Huntington's disease. What do we know? We know a great plenty. From 1872, when George Huntington described the illness as a family disease, and the hallmarks and advances that we've made, we know a great deal about this disease. I'm not going to go into those today because frankly, you're the audience that has helped the scientific community define this for healthcare today. I just want to point out some of the pioneers in our field, the people that first identified the marker for the disease, and then were part of the HD collaborative group that identified the gene and changed the entire face of following people with an autosomal dominant genetic disorder. And this is Jim Gazzella and Marcy McDonald, who continue to be our warriors for understanding the genetics of Huntington's disease. I also need to thank the first person who kind of reached out to the families and said, okay, it's great that we have this in labs and sciences, but we also need to reach out to people. And Nancy Wexler reached not only across the aisle in this case, but across borders to bring in families in Venezuela and worldwide to help us learn more about this disease. And I can't tell you what a difference it's made. I've been able to witness it. The more involvement we get from the families, the better our science is and the more rapid we can advance it. And then I need to thank the first person, at least in the United States, the first person who built a network of researchers. Ira Scholzen and his team put together people and said, look, we all are in this together. Let's build the Huntington Study Group and put similar researchers together so we can share and build things faster instead of working singly or in silos. So I think this team, as well as many, many, many others, have really made a difference. But these are three important points I want us to think about, and it all comes back to teamwork and partnership. Whoops. The next question the FDA asks us is, do you really know how to diagnose this disease? Do you know when it's there? And frankly, we have a little easier job because it's a genetic disease. We've known for years that it was a family-based disease. We've known for 2 or 3 decades that we can identify where the gene is, and more recently, exactly what the gene mutation is. And then through the efforts of all these team scientists hard at work around the world, we also know what the stages of the disease look like. Now, this is a slide I'm going to move over with some rapidity today, because this continues to open up how we identify and diagnose disease is an ongoing question. It isn't done once and then you're done. We have to continue to integrate the scientific findings into our diagnostic criteria. And there's a lot of discussion going on today, which is so exciting, about how can we better integrate the scientific knowledge with the clinical application of that knowledge. So this slide in the upper right-hand corner just shows these 3 stages: red, green, blue, 1, 2, 3. And this is, you know, the data from PREDICT. It's old data at this point, but it shows that we were easily able to identify multiple measures that disease was starting much earlier than the motor diagnosis was given. And I think this idea of continuously improving our state of the state has occurred in the most recent publication that I'm aware of. Actually, there's been multiple, and I hate to highlight one over another, but CHDI just put together a group that used data. Whoops, I'm going to go back. I think I bumped it. Maybe. There we go. So CHDI just put together a new publication, and they are trying to help us stage Huntington's disease. And now this is going to be an ongoing work, and I think we all need to be involved, not just the scientists, not just CHDI, not just one university or one country, but worldwide. Families, scientists, everyone needs to dialogue about how we can keep science and clinical care working together. This is the Huntington's disease integrated staging system that's been proposed. And it's been proposed using an amazing amount of shared data, which I will show you as well. And this is the latest for how do we diagnose, how do we stage illness, and should we really be using the same metric on clinical care and research? And this is going to continue to evolve. So I do want you to be aware of it and get involved. This slide is very busy, and you don't have to be able to interpret it, but this goes back to the FDA's question of, will people volunteer? Now, when we got PREDICT funded in the first year, everyone said, no, no one would volunteer. And I'm so pleased that we were wrong, because they said you won't even get your sample size. The first grant went in and wanted to find 400 people who would volunteer. And at the end of the study, we had 1,500 people volunteering. And my talk today is to say we can't stop these explorations and this advance of knowledge that we've been working on. This slide, however, I want to direct your attention to the upper right bar chart that shows green, red, purple, and blue bar charts. And it just shows how many people have come forth that are willing to volunteer their time and valuable energy to work together in partnership for us to learn more about Huntington's disease. And you can see in this slide, there's literally tens of thousands of people volunteering. And this data is now shared worldwide. And many people have asked me, well, now that you've got all that data, why can't we just, just run the clinical trials and fix everything? Well, that's why we're here today. And maybe this will be the first of several, but there's plenty left to do. And my whole message today will be, let's not slow up, let's keep moving forward. And there's plenty left to do. And I think HDSA does a good job of keeping us on our toes with the new things that are coming on board. So when FDA asks, do you know what you're doing? Do you know how to diagnose? Do you have volunteers? I think we can say yes to all three of those easily. And then when they come back, because they will, and say, are you sure? Because everyone has to doubt each other. That's who we are. And I think we can answer, yes, we are sure. And that's what today's about. It's getting us back together as a team, getting us back together to realize what we can do as a joined force. Oh, the slides are just having their own mind. That's fine. So these are the requirements. I think we can thumbs up. We've taken care of all these. So my question today is, what's next? I canvassed the internet, as we all do regularly, and asked about prevention. This is a big challenge, and it's a challenge that we are not the only ones that are looking at earlier ways to look at illness and try to maximize wellness more strongly, earlier, longer. And this is a new challenge for healthcare. And this is from a really brief sampling, because I didn't want to take that much time on it, of the number of organizations and groups that have gotten together to say, let's not just treat arthritis and diabetes and Alzheimer's and Huntington's. Let's try to prevent it. Let's figure out how we can get our interventions to be earlier so people can still enjoy their lives, raising their kids, driving their car, keeping their jobs, and fulfillment of any life that they choose. So this is just to tell us we're not alone. We're all working together. There's challenges out there, but there's people we can learn from. So we're going to look specifically at prevention of Huntington's disease. What do we need to do next? We started this decades ago and we've learned a lot, but we're not done. And I think it's easy to say, oh, PredictHD is over and all those studies, we learned a lot, but we're done with that. We're on to the next thing. And my call today is that we're not done. We need volunteers, we need partnership, we need new ideas, we need good scientists, we need everyone to continue to work together so that we truly can get to a stage of our disease where we can treat it at any stage. We can treat it in late stage when we provide care. We can treat it at early diagnostic stages where we can maximize how someone can function. And we can treat it before diagnosis to keep them functioning as highly as possible. And maybe eventually we'll even be able to push the age of onset back so that people can get sick at a later age in life. So we have our goals set up for us, and I know we can do it. So let's get going. So a lot of times people say, what's next? What are our choices? And I love the, two roads diverging in a wood, but it's not two for us. There's a lot to do. So I'm just going to put it in context that if you feel like, well, that study's not for me, or I really don't want to do that study, or no one in my family fits that criteria, there's plenty for everyone. And we really need you to step up and reach out to us and say, what can I do to be involved in making a difference for the lives of today's families and tomorrow's families with Huntington's disease? So I'm going to go to our newer questions that we don't have agreement on, and it's okay. Of course we don't have agreement. It's new and these need to be worked out. And I'm not saying we should force an answer to anything, but I am going to ask us two pretty important questions that we don't have the answers to. The first one is who, meaning what type of people should test our new treatments. And every clinical trial to date, with 1 to 2 exceptions, I think there are 2, are in people that have manifest motor diagnosed Huntington's disease. There have been at least 2 trials that I'm aware of that also brought in people that were at risk. But it isn't the state of the art, and the FDA is challenging us to make sure we take care of the missing pieces and fill the gaps in our knowledge base. And that's what I think PREVENTHD is going to be all about. We got to fill the gaps of what we don't know regarding what is the natural history of disease. How do we make these decisions? Like, who should the treatment be offered to? Is it going to be one treatment fits all and it's just going to be a pill you take, and or maybe it's a one-time gene therapy that we do at birth for everyone that has a gene expansion, or maybe we wait till adulthood so they can choose for themselves. But really, that's unlikely. It's much more likely that we're going to have to weigh many components of the decision of which treatment is best for me at this time. So these have all been proposed. Right now, of course, the giant winner is we go with diagnosed Huntington's, and it makes the most scientific sense and fiscal sense and resource sense because we know how to measure it, as I shared with you. But there are a couple studies that have gone with just bringing in everyone at risk, so they don't have to know their gene finding if they don't want to know. We have studies that said, oh, it's too expensive to spend all the extra money in the technologies we do today. So we really want to at least only do it in people with known genetic risk. More recently, and this includes the paper by CHDI, they're supporting that we only do it in people with a 40 CAG repeat length or higher, so that we don't have people that are slower to onset or may not be fully penetrant. And others think we should just limit the CAG repeat range completely. And this isn't a decision that one person makes. Every study, every treatment will have to make these decisions, but they shouldn't be making a decision without your input, because we need to hear from the families, from the stakeholders, from all the different scientists, all the different countries, all the different people around the world. How are we going to do this? Now, I am encouraging you to be part of that team to discover how we're going to address to provide the right treatment at the right time in the right person to maximize your life. My daughter's generation is constantly encouraging everyone to live your best life. And I think that's what we're all doing when we are trying to take whatever we've been given, whether it's a gene for something or the multitude of other challenges that life can give us. But we want to see what we can do with it and how can we live our best life. So this is what we're trying to do with PREVENT. We're going to take all these gaps in our knowledge base and try to fill them in so that we have answers to these things, not for the health professionals to make the decision, but for families to say, okay, this is my choice. Personally, I'm from a cancer family, and there are lots of decision-making points in cancer. You know, if you get a brain tumor, depending on what type it is, in your 20s, they may just take out the tumor and then wait and watch. So that's one approach, but there are many approaches. They also don't know when to start with the heavy-duty chemo or radiation that might have other significant side effects. So this is something that we as a community need to start thinking about. These are like how we would choose who we would enroll in the child trials and people's opinions. Everyone has an opinion and they're all different, which is beautiful because we want all points. Some people think diagnosed is too late. Some people think we can't look at everybody at risk because it's too expensive. Some people think we shouldn't just do gene carriers because it might be coercive or unethical if we force everyone to get tested. And it just fits perfect with our call from Dr. Johnson today that we need to call for this Parity Act. It isn't a safe culture yet for us to be open about our genetic risk. We have often been the first disease to pilot something, and we want to be safe. We want to take care of one another. So we also need to figure out what is the best answer for this. Yes, if we only go with fully penetrant numbers of CAG repeat length, that might make the clinical trial faster, but is it equitable? And I think in our life today, we're really being challenged to say, are you being fair? Are you allowing diversity in? And are you being equitable with the diversity that you have. And this isn't something we've talked about as a field before. So I think we need to continue with this mindset. The last one here is that some people think we should just treat everyone at birth so that it's done with. But the reason I want you to think about that is because it's going to depend on the treatment. Some of the treatments today have brain surgery. The side effects could be very large. Some of the treatments today are taking a pill that the side effects may be a little nausea, headache, vomiting, maybe dizziness when you first start it. These are hugely different treatments. And some of them, if it's an infusion in your spinal column, maybe you can do infusions for 10 years, but can you do them for 40 or 50 years? We don't know these answers. So I think as a community, we need to join together to answer these questions. So the second big question, the first one is, who are we looking at? The second one is, how are we going to know if a treatment works in a group of people that don't really have the diagnosis yet? We can't use the tried and true scales that were developed, what now we can call the olden days, because they're only for people with manifest disease who are losing their ability to feed themselves, to take care of themselves, and to be independent. So now we need to look at a bigger picture. What measures can we look at? And there's lots of ways to look at this, and I don't want to spend too much time on it, but the most recent ones that are really pressing at this point is there's different opinions on what are our best biological markers. And this is the far up right corner where you see a picture of the brain, the MRI scanner, picture of a person undergoing a lumbar puncture, which is the needle in the spinal cord, and then and then the arm where you just draw blood. So we need biological markers of disease. Of course, you say, well, we have a gene. Well, yes, we have a gene, but we know that people live long, wonderful lives with that gene until they start to have impairment. And that's what we need to identify. So we need to really start working together on these. What's starting to happen in our field a little bit is we have all of these answers to what's the best biological measure, but we don't have consensus. So much needs to be done. So the PREVENTHD study really encourages people to try to get all the measures in the same people. The phenotype, which is the measurement of how you're feeling, functioning, thinking. The biologicals in your blood, the biologicals in your cerebral spinal fluid, and your brain pictures, no matter how we can take those, whether it's through PET or MRI or new technologies that haven't been developed. I really think we need to all step up and try to get these databases to be as complete as possible. The next way we measure things, I'm going to quickly go through these because you're familiar with them, is clinician report. We get experts in and look at us and say, well, which one of these? And you all are familiar with finger tapping or chair stand test or eye movements. We also have you take neuropsychological tests, the best friend of me. I really like these objective tests that you can take, and then we don't really have to rely on a number in your blood that says, yes, you have this number. Instead, we can look at within you. Has it changed over time? Because there's no reason you should be going backwards in your ability to function and think and feel. And if you do decline in these areas, we really need to pay attention. So I really like these objective clinical outcomes. I've thrown some up on the screen with our finger tapping, our emotion recognition, our block designs, and even our smell tests. The other thing we learned in PREDICT is that it is helpful to have other people's reports. Because it turns out in our PREDICT, which means people that were not diagnosed, about a third of them had a disconnection of their circuitry that gave themselves feedback. So they really couldn't tell how they were doing. So I think it's best if we can design studies that have this input from your family. Now, I know we all go, well, I don't really want to ask my husband or daughter if they think I'm irritable, because you know what they'll say. Even on my best days, my adolescent kids are going to always say I'm irritable, right? So let's figure out a way to do this to get the best measures in all the time. And the last thing and the most important thing on this slide is really asking the families, you who carry the gene, what do you witness? What do you get feedback about? What are you struggling with that maybe we're not even measuring yet? And most of our best measures have come from the families. So this is really, to me, the most important part is we need to ask you, and you need to stop and think, what is difficult for me? What do I do well, but what am I having trouble with? So what other fields are doing in this preclinical, prodromal, before diagnosis, preventive era is they're trying to stack up all the measures of how you're functioning, all the measures of how you feel, all the measures of brain measures. And this slide is from the Alzheimer group, where they're comparing maybe shape changes in the brain to PET changes in the brain to MRI changes in the brain to little measures like how do you name things rapidly, or how do you remember things or learn new things, memory, concentration, what is your language like? They're putting it all together and they're trying to pick what's the best measure. Now we've started to do this, but really I'm making a call that we need to get back to our teamwork premise of working together to do this, because what's happening now is we're a little bit siloed. We have a study over here that says this is the best measure, and a study over here that says this is the best measure. And what they mean when they talk about silos, you can see lots of graphics on the internet, is when work is done without communicating across countries, across disciplines, across sites. And I'm really encouraging us to get out of our silos again and share the way we did in the past so that we can really get a handle on what's our best biomarkers, what's our best clinical outcome measures. So the PREVENT study is really an argument that we get back to this before diagnosis group. We get as many volunteers as possible. And what we're looking for are people that have been gene tested and carry a mutation in the Huntington's gene, or don't. We take both because we need to compare how you're doing across the board. We need to say, okay, if everybody kind of gets some stress in midlife and their cognition takes a dip, or you get depressed at adolescence, is that normal, like your siblings that don't have the gene, or is this an early sign? So that's why we need lots of volunteers. And for this study, I really am a believer in trying to do everything in our cohort. So we're doing a lumbar puncture, which is trying to get a sample of your cerebral spinal fluid. We've successfully done hundreds in PREDICT, and we've already done 50 in PREVENT. And I really think it's going to be important. This data will be shared worldwide for people to say, I have a new idea for a biomarker. I'm going to look at the inflammatory pathway, or I'm going to look at the immune pathway, or I'm going to look at the allergen pathway, or, there's so many things to look at that we're really only looking at a very few. So we also are going to do brain scans and ask you every question we can think of. So here's contact information. Feel free to reach out. I carry a separate little cell phone just for HD, so you can text me. If I don't pick up, it's because I'm giving a talk like now. I did mute this for now. But please reach out, and I will put you in touch with whatever study is best and needs your help, because we really need to work on team science. In my opinion, it takes a village, and we have a village. So let's be that answer to a better future for everyone with Huntington's disease. Thanks, I'll stop there.
Dr. Paulsen.
Thank you.
Yeah, thank you. Sorry to jump over you there, Kelly, but I just wanted to say, Jane, it's always good to hear how you champion the involvement of the patients and families in HD research, and how important it is that that voice is heard, because as you've said, the complexity of HD research is something that it's hard to wrap our brains around, right? There's so many different things being looked at and so many different pieces in motion at all times, but to remember that it keeps coming back to the people who are living with HD that are driving these things, both the studies themselves and motivating the people who are doing the work to keep doing it. So thank you for that.
Thanks, Eric.
Thank you. And with that, I think we can get started on our Q&A. We've had some good questions roll throughout the talk, but I think a good place to start might be, can you clarify the differences between PredictHD and PREVENTHD? And if someone participated in PredictHD, are they also eligible to participate in PREVENTHD?
Oh, certainly. And I think that's one of the first things we need to do. And I want, at least from my vantage point, I have reached out to all other studies that are recruiting the same type of people, these before diagnosis people, and I can directly discuss how we have shared motives. And like, for example, I know the other study that I really want to keep moving right now is the SHIELD study, right? And they are seeing people every 4 months for over 2 years, and they're doing a largely prior to diagnosis sample to really get better measures. And I think this has to be done as well. And the cool thing about that study is the triplet repeat group are specifically doing this with an idea of a disease modification in mind. That's the way I see us moving forward. It's sharing and being open with one another. And every data point that we have collected and predict, we make it available as after we assure that it is safe to do so. It's been cleaned, it's been tested to make sure it's good. We make all the scans available. And the only difference between the two studies is PREDICT literally was funded from 2000 to 2014, and then it was funded from what's called ancillary grants through 2017. So all these added on people we invited to say, you've got a cool technology or you've got a cool expertise. Can you look at PREDICT? So from 14 to 17, all these other scientists were working on PREDICT. Many of them were recruited to Huntington's disease for the first time. That was a very exciting time. And then at the end of that time, we were hoping that everyone from PREDICT would go in to enroll and we could continue the focus on pre-diagnosed HD. Also, however, we knew by 2017 when we concluded PREDICT that we really didn't have the measures we needed to enroll. So we need you to be in enroll. It's a great place to get everybody in there, but it doesn't have the level of sophisticated science that we have in some of our more detailed studies, like a brain scan or a lumbar puncture or the blood draw or the biomarkers. So the only difference between PREVENT and PREDICT is that we're the upgraded next gen 2.0, you know, we're trying to take all that knowledge that this team has helped us gain, and we're saying, okay, what can we do better? Because we really believe that we learned a lot, but we're not done. I'll give you an example. One thing that we're not done with is we're seeing people once a year. For most dementias, that works fine, once a year, right? And even our other studies, we see people once a year. But that's why I'm really excited, because when it gets to a clinical trial, you're not going to see someone once a year and hope you catch the little window of change. So I am really interested in trying to figure out how we're going to measure things more frequently. And I'm not alone. There's people around the world in Huntington's research who are looking at wearables and at-home systems and doing it on your cell phone or logging into the web and collecting more measures, because we know that the more measures we get from people, the more able we're going to be able to see the very subtle changes that occur with this disease over time. So that's one thing we've done, is we've adapted our tasks all to iPads during COVID. The good news is we shipped them out and did them from home. We just kept our visits going. The bad news is now our methods are all messed up, because that whole year was iPads from home, and now we're doing it in person again. So there's always an up and a down with our challenges in science, but we really need you back. And I need enough people that are willing to come in and do it in person. And then I'll mail you an iPad and we'll do it again, because we need to know, are these going to be good measures? Because I think they are. And I think they'll be better measures, because we all know we have good and bad days. And that day you go to the doctor, it's kind of like when you take your car into the mechanic. The problem you've been having for 3 weeks doesn't happen in front of the mechanic. You know, and he looks at you like, oh, crazy lady. But really, it just doesn't happen that way. So we can't trust that the one visit we have every year with our neurologist, we're going to capture everything, what it's like to slowly have changes occurring in our brain. So I'm really excited about that change. And we have lots of changes like that throughout the study that just take what we know and add a piece. The grant was funded for biomarkers to test 5 biomarkers. At this point, some of those biomarkers we already know aren't useful. The grant was funded in 2017. We found out last year that some of those outcomes people were banking on really don't show the variation and change over time that we needed them to. So we really need the science to continue. Instead of saying, oh, we're done with everything, we're just going to take the next treatment. We need to keep the science going so we have the best measures. So that's the other thing we're looking at, is we're really trying to stay on the cutting edge of what is the best imaging measure, what's the best CSF measure, what's the best blood measure, and what do you notice in your day-to-day lives. So I need to stop because I'll talk forever. Thank you. Thanks, Derek. Does that help a little?
Yes, absolutely. Yes, all very useful information. And I love hearing about the COVID adjustments too. It's always, a challenge to see how do we continue in research in this new era, but that is very awesome to hear. Thank you for sharing. Let's see, can you clarify, are individuals at risk for Huntington's disease eligible to participate in this study as well, or is it exclusively people diagnosed with HD?
This one is at risk, and it is a little bit of to me, a little old-fashioned, because we're still going back to the you must have been tested. I did try in my more recent grants to get it funded like some of the other studies have. DIAN, which is the Dominantly Inherited Alzheimer's Network. They'll just test you in the study and you don't even have to know, and you can still volunteer. I know they will approve that for us, but let's face it, we were the first. And when we went in, we had to be super cautious. It's just like we're the people that wrote that GINA bill, which is the Non-Discrimination Act for Genetic Information. We don't want to take a risk and have something not be safe. So in a way, that's the good and the bad of being first. The good and the bad is that we're super cautious, and other diseases are now able to just bring everybody in and get it paid for. But our grants still require us to know up front. So we're working on that, and I think we'll come to some good successes of being able to bring people in so that you don't have to know, so you can still have the ability. We want to empower people to make your own choices. When do you want to have the brain surgery or the infusions or the injections or just the pill? That's for you to decide. No one can tell you what to do in the long run. So I just will say that there's a lot of work to do there. I lost my train of thought. What else did you ask?
No, that was great. Thank you. Yes, just kind of who is eligible for the study. And another question we had come through was to clarify on one of your slides, you said fully penetrant individuals are eligible. Can you clarify what fully penetrant means?
Bring it up is because it is new. There's a lot of, it's really exciting. It's a great time to get your voice heard because people are not in agreement with how we define what is Huntington's, which is at some level a little shocking because we've known for hundreds of years what Huntington's is. But on the other hand, that's how much we've learned. We've learned so much that now people say, okay, well, we know it's 36 or above CAG repeat length, you can manifest Huntington's disease. Lower than that, you can give the Huntington's disease on to an offspring, but you probably won't manifest it yourself if you're below the 36. But now this new CHDI paper is saying, well, because they don't always manifest it as predictable as those with 40 and above CAG repeat length, maybe we should only be researching the people with a 40 and above. So everyone has an opinion on this. There's good and bad news. I tried to give you a little sprinkle of it in the talk of the opinions about all these, but you need to weigh in because it's going to be different for everyone, just like it is for any disease. Young diabetics are different than old diabetics, and diabetics that are overweight are different than diabetics who aren't overweight, and diabetics with big vascular component are different than the diabetics that don't have that additional heart disease. So we have to do the same. I really want to include everyone, but for the most part, I'm trying to take the pre-diagnosed and follow them over time. Now, right now, I do need anyone who gave a lumbar puncture in PREDICT to come back because this grant is funded based on the PREDICT data that we captured. So if you had a lumbar puncture in PREDICT and you only had one, we need you back for PREVENT, because they're going to pay for us to look at more biomarkers. So then we can look at more things in the CSF and more things in the blood to see what's changing. Because the one-time thing is much more difficult to tell what's going on versus in the same person getting the same measure over time. So spread the word, we need PREDICT back even if you're diagnosed or impaired, if you've had one lumbar puncture, we need you back for a second. Other than that, we're trying to just take new people who are willing to step up and help us develop these new tools.
Hey, Jane, I think there's a couple of practical questions that came up related to that. Someone had asked if the lumbar puncture and other biomarker sample collection, is that only being done at the University of Wisconsin, or are they able to do that at other sites?
Right now, this PREVENT study is only done at one site. To be honest, NIH did approve me to do a multi-site study. But given that all the challenges that happened during this time frame, it was funded in 2017, so started in 18, we also had shutting down 33 PREDICT sites, and we also have had some real excitement going on in clinical trials, and I really felt like our community was putting their energies where they needed to. So I only am doing PREVENT at one site, but I do hope that we can continue this pre-manifest cohort, hopefully at a few more sites, not 33. Oh my gosh, that was way too much work, but thank you for participating because it really helped us learn. But I don't think we're going to get there. Enroll is the study to get the massive numbers, but it isn't going to have these details. So one thing we do, we are working with everyone, not just Shield, we're working with CHDI. They're happy to share their enroll data so we don't have to give the test twice, but then we have to do the lumbar puncture and the brain scan. And my goal is to get this at a few more sites in the future, but right now I'm only doing it in Wisconsin, and partly that is still a COVID response. It's been very tricky to launch science during this pandemic when we don't know what is going on with travel or safe practices. So I just want to thank everybody again. The people who are coming out during COVID time to participate in Huntington's research when it could be extra. Well, it is. It's just extra exposure. So we're very appreciative. We don't want research to slow or stop, but we also want to put you first. I want everyone to put yourself first. And for now, it's just going to be at one site till we figure out what's going on in our communities. I'm open to your input on that.
You know, someone had asked this earlier, and I'll ask it now. Is it possible to put the contact information that you shared on your slide into the chat so people can capture that and get in touch?
Definitely.
While we're still on, if someone can do that, that'd be
team members here. Tricia Denman is the new program manager for all the new studies that we're putting together. And if she could put it into the chat, we can definitely do that, because we want you to have we have a specific email, we have a specific phone number where you can call or text, and you can always just email me as well. I put that up on the shared information, and we'll get back to you no matter what. It doesn't matter if you're not eligible for this study. I want to get you somewhere. If you're willing to volunteer, we need you. I'll call Eric back up, or Kelly, or Leora, someone else, and say, this is somebody who's willing to volunteer, you know, with their kids or with their parents or with their siblings. And there's so much cool stuff going on right now. We really need everyone to participate, and I will help get you where you need to go.
Someone asked, are people who know that who have tested negative, are they able to participate in this study? If you're looking for
That's one of the problems from our data was they said we didn't have enough negatives. So what good is our biomarker research if in PREDICT, which comes back to me, this is my fault. I was so focused on getting the gene expansion carriers to follow over time, because frankly, everyone in PREDICT at some level looked perfectly normal and healthy, right? Because we were following them over time. So I didn't place an emphasis on the negatives. So now when they look at our data and our samples that we're going to analyze for biomarkers, they say, you need more comparison samples. So yes, we are definitely seeing people that tested but don't have the gene. So we're really, the question was good that you asked, but the answer is not a good answer, because it's yes, we're seeing everybody. We're seeing people diagnosed if they were in PREDICT and had a lung puncture. We're seeing negatives if they can fill our criteria of helping our sample size for the FDA be better balanced. But primarily, we're still looking for those early people who have been gene tested, so we can follow you over time and figure out what's the first CSF marker, what's the first blood marker, what's the first smell deficit, what's the first brain imaging deficit, and you tell me what's going to happen next. And it looks like the contact information was shared. I hope, is it all there? Because I don't have that part open.
I think we just threw the website in, so it should all be there.
Thank you very much. I appreciate it.
No problem. Let's see, have we looked at, so the biomarkers that are being looked at in this study, are they specific to Huntington's disease, or are they also applicable to other neurodegenerative disorders?
Both. What we have done in our previous work, because this isn't our first biomarker project, is the grant was written with whatever looked important at that time, which, as you know, our biomarkers have changed over time. It was written for neurofilament light chain because we already knew that looks like that is going to be good for any neurodegenerative disease, but probably not specific in that it goes down in a lot of diseases. So, but still, if that's going to be a good biomarker, we need to know when it hits the level where we should intervene with the treatment. That's the information we need to do. So there have been some studies that have shown that the imaging is completely normal when the neurofilament light starts to go down. Unfortunately, because we're still working in these silos, we can't compare across studies yet. So I think we have, when you work with people, just say, are you sharing your data, and can we get the data put together and share it? The CPATH Institute is the HD Regulatory Science Consortium, and they really would need everyone's data so we can compare these answers across studies. And that really isn't, we're not able to do that yet because not everyone is shared. So I think sharing is first. We were also funded to look at the mutant Huntington protein. We're also funded to look at inflammatory markers. We're also funded to look at, like, I think it was 5 to 7 we were funded, but some of those, someone else, Ed Wild or his group or some other group, CHGI, has looked at them, and I don't want to waste the resources we have because we only have so much money. So I'm open to whatever you think we should be looking at. I still have several in our list that we're going to test for. And I'm open to new ones if you know a scientist or if you want to recommend your favorite biomarker.
Awesome. All right, we are coming up on our time here, so I'll just go ahead and ask one more question for you. You've mentioned that there are other observational studies. Can you talk to us a little bit about the importance of observational research as we're continuing to learn more about HD and how all of these different observational studies can complement each other?
Yeah, I think it's going to be important that we set up a structure like we did many years ago when PREDICT occurred. We kind of got everyone on board working towards the same goals. Now, the good thing is we still can appreciate everyone doing their own thing, because every site that it's a PREDICT site, they could still go do their own thing in addition to doing PREDICT. But what's happening now is people are setting up a study over here and a study over here. And if we don't share the data, we got apples and oranges and we can't compare them. So we really need people that are willing to share and team science and work together, and that are really in it for making progress for the families with Huntington's disease. And that's really the main thing to me. And yes, we're working with everyone possible, and Roll is willing to share their data. As I said, Triplet is sharing their data. They're the only two. Oh, there's a YAS young adult study in Europe, but I'm not recruiting in Europe. I'm having the Europeans there, and our continent is here. Partly, again, it's travel. We can't really deal with cross-country things right now. So I think I have reached out to the other studies in Europe and around the world, in Australia, and I will continue to do so, because I will share this data with everyone, and I want to compare their data with our data so we get a good answer of when neurofilament goes down and when the brain imaging measure is best. So I don't think we can do it if we continue to work in silos.
Great. Well, look, Jane, thank you so much, Dr. Paulsen, for making the time for us today and for sharing everything you did. For everyone who attended, thank you for making the time today and for the questions that you provided. Again, the information for Dr. Paulsen's study and contact information was shared here in the chat, but we'll also be sharing the entire presentation online. It'll be documented on our website, hdsa.org, as well as the YouTube channel. So please check it out later or share it with others who weren't able to make it today. Thank you so much to everyone, and we wish you all the best and look forward to seeing you in the very near future at another webinar.
Thanks.
Take care.
You too. Take care.
Thank you, everyone.